Most enzymes are active only within a very narrow pH range. The cytoplasm of the cell has a pH of 7, and most enzymes function best at this pH. A plot of reaction rate versus pH for a typical enzyme is shown here below. So you can see that the rate of reaction increases as we increase the pH until we get to this optimum pH. This is where the rate of reaction is maximized, and then as we increase the pH, it drops off. The pH at which the enzyme functions optimally, we call that the pH optimum. Making the solution more basic or more acidic sharply decreases the rate of reaction. These pH changes alter the degree of ionization of amino acid R groups in the protein, as well as the extent to which they can hydrogen bond. This causes the enzyme to lose its biological active conformation and it becomes denatured. Some environments within the body must function at a pH far from 7. For instance, the pH of the stomach is approximately 2 as a result of the secretion of hydrochloric acid by the cells of the stomach lining. The proteolytic digestive enzyme pepsin must effectively degrade proteins at this extreme pH. In the case of pepsin, the enzyme has evolved an amino acid sequence that can maintain its tertiary structure at this low pH, and is most active in the hydrolysis of peptides that have already been denatured by a very low pH. Thus, pepsin has a pH optimum of 2. In a similar fashion, another proteolytic enzyme, trypsin, functions under the conditions of higher pH found in the intestine. Both pepsin and trypsin cleave peptide bonds by virtually identical mechanisms, yet their amino acid sequences have evolved so that they are stable and active in very different environments. The enzymes in our cells are rapidly destroyed if the temperature of their environment rises much above 37 degrees Celsius, but they remain stable at much lower temperatures. This is why enzymes used for clinical assays are stored in refrigerators. The figures on the right show the effects of temperature on enzyme-catalyzed and uncatalyzed reactions. The rate of uncatalyzed reactions steadily increases with increasing temperature because more collisions occur with sufficient energy to overcome the energy barrier for the reaction. The rate of an enzyme-catalyzed reaction also increases with modest increases in temperature because there are an increasing number of collisions between the enzyme and the substrate. At the temperature optimum, the enzyme is functioning optimally and the rate of reaction is maximal. Above this temperature, increasing temperature begins to increase the vibrational energy of the bonds within the enzyme. Eventually, so many bonds and weak interaction are disrupted that the enzyme becomes denatured and the reaction stops.